Before we move on to the pathology section of the chapter, we have to review just a few more basic concepts relevant to the anatomy and physiology of the musculoskeletal system. One of these topics is the process of muscle contraction and the conduction of electrical signals that lead up to it. What are the basic cellular and molecular components of muscle contraction? Let's start with the cellular signal conduction and receptors. In an individual myocyte, this invagination represents a T-tubule. On the surface of the cells, you have dihydropyridine receptors, which are voltage-gated calcium channels. Within the cell, lining the sarcoplasmic reticulum, you have ryanodine receptors, which are calcium-induced calcium channels. So you have two receptors set up that manage calcium. What does the SR and its channels do? The SR stores calcium, and the channels control calcium flow. Calcium, as we're about to get into, is integral to the process of muscle contraction. Here you see the structure of a muscle cell, with multiple myofibrils. You can think of muscle fascicles as made of muscle fibers, which are themselves made of myofibers. What are myofibers comprised of? Sarcomeres, which are the smallest functional units of muscle cells. The different bands, zones, and lines detailed in the picture of the sarcomere are worth looking at. Let's spend some time on the sarcomere, because this will definitely appear on your step 1. In general, the sarcomere extends from Z-line to Z-line. You can remember that Z is at the end of the sarcomere and also at the end of the alphabet. Off of the Z-line, actin molecules project in towards the M-line. Off the M-line, you'll notice thicker lines. What are these thicker lines? Myosin that is extending towards the Z-lines. The thin actin fibers and thick myosin fibers overlap, always at least to some degree. The M-line is simply the midline of the myosin chains. The H-zone represents the part of the myosin chains that do not overlap with actin. The I-band represents the part of the actin chains that do not overlap with myosin. Where is the H-zone the longest? In relaxation. Finally, the A-band is the length of the entire myosin chain from end to end. Does the A-band change with contraction? No, the A-band is always the same. Alright, take a look at this contraction. The I-band and the H-zone will get smaller with contraction, as the area of overlap between the actin and myosin increases. In relaxation, the I-band and the H-zone elongate. In summary, the H-zone contains myosin only, the I-band contains actin only, and the A-band contains myosin and the myosin-actin overlap region. The Z-line is the site of actin attachment, and the M-line is the site of myosin attachment. To remember the distinction, you can use the memory aid His shrinkage, and the A-band is always the same. The way this will appear on your exam is to look at an electron microscope image of the sarcomere. Here are the Z-lines. Can you match up the other pieces? This is the M-line. You can also see the H-zone and the I-band. If that didn't make sense, we'll review these concepts as we work through the steps of muscle contraction. The first thing that begins all muscle contractions is an action potential depolarization, which then travels down the neuron, opens voltage-gated calcium channels presynaptically, and causes neurotransmitter release. Once the neurotransmitter reaches the postsynaptic neuron, the muscle cell begins depolarizing at the motor end plate, which spreads by voltage-gated sodium channels. This depolarization will travel down the muscle cell and into the T-tubule. Remember this picture from before? This causes depolarization of the voltage-sensitive dihydropyridine receptors, which allows calcium to flow into the cell. This calcium causes a conformational change in the ryanodine receptor on the intracellular SR. This leads to a conformational change that causes calcium to be released from the SR. Calcium was released from the SR because calcium entered from outside the cell. So what's with the high level of calcium? This is an actin molecule from the sarcomere. Tropomyosin and troponin molecules are associated with these actin molecules and act as placeholders. Calcium binds to troponin C, think C for calcium, which then causes a conformational change, causing tropomyosin to move out of the myosin binding groove on the actin. You can remember that tropomyosin is the adapting molecule between the calcium binding troponin and the contraction generating myosin portion by keeping in mind that the name incorporates both sides. What is this molecule? It's myosin. In response to the movement of tropomyosin, myosin then releases prior bound ADP molecules and is displaced on the actin filament, the so-called power stroke, which is actually how myosin molecules move along actin. Do you know why bodies develop rigor mortis after death? It's because after death, ATP generation ceases, and there's no more ATP available to bind to the myosin and release the cross bridges. ATP binding is critical for the detachment of the myosin head from the actin filament. There is an important distinction between skeletal and cardiac muscle as far as the basic structure is concerned. Skeletal muscle is composed of one T-tubule plus two terminal cisternae of the sarcoplasmic reticulum, called a triad. It's like a sandwich of membranes, where the SR is on either side of the T-tubule, which is sandwiched between them. These are at the AI junction. Cardiac muscle, by comparison, is comprised of dyads, or one T-tubule and one terminal cisternae. It's located at the sarcomere Z-line and allows for excitation-contraction coupling. Let's have a flash quiz. What part of the sarcomere does not change length with contraction? The A-band. It is always the same. Before we go further into the nitty-gritty of the power stroke, are there different types of muscle fibers? Yes, there are two. This is a cytochrome oxidase stain, where the type 1 fibers stain dark and the type 2 stain light. You should think about type 1 fibers as being responsible for sustained contraction, while type 2 fibers are responsible for fast twitch. Type 1 fibers are slow twitch, and you can remember everything you need to know about them with the memory aid 1 slow red ox. That is, they are slow twitch, they are red fibers, meaning they have a lot of mitochondria and myoglobin, and ox, which means they utilize the full oxidative phosphorylation pathway in their mitochondria. Conversely, which metabolic pathway, glycolysis or oxidative phosphorylation, would the white fibers rely more on? If you said glycolysis, you're right. As you'll remember from the biochem chapter, glycolysis is much less efficient and requires a significantly higher amount of glucose input per ATP generated than oxidative phosphorylation. Which muscles are slow twitch? Slow twitch muscles tend to be the postural muscles, for example, your paraspinal muscles. These muscles have long sustained contractions. Just think about when you're walking or just sitting up straight. This allows ample mitochondria to have time to go through oxidative phosphorylation and create enough ATP to continually power these muscles. In contrast, fast twitch muscles are white because they do not have a lot of mitochondria or myoglobin. They primarily use anaerobic glycolysis. Fast twitch muscles include muscles like your biceps or your extraocular muscles. These muscles can't wait for additional ATP to be generated through oxidative phosphorylation. They need ATP and fast. This muscle is also the type of muscle that commonly hypertrophies in weight training. As promised, we're now going to return to the topic of muscle contraction. Before we were looking at the sarcomere and its greater organizational structure within muscle fibers and even muscle fascicles. Here, we'll look within the sarcomere to see why the areas of actin and myosin overlap and why they actually contract. The basic idea is that the heads from the myosin chains attach to actin and stroke along them, a process that's dependent on ATP. You can think about the first step as a myosin head that's ready to attach to actin. In the resting state, that myosin head can't attach because tropomyosin blocks binding. However, when the signal for muscle contraction comes, calcium rushes into the cell. And where does it come from? The sarcoplasmic reticulum. And where does it bind? To troponin C, which causes the troponin C to shift out of the way. This reveals the myosin binding site on actin. The myosin is already in the cocked position, so the next step is for the myosin head to power stroke along the actin, which results in the loss of a phosphate group. Next, ADP falls off the myosin head as well. This is what triggers the power stroke, causing the actin and myosin fibers to slide past one another and cause contraction. Now is the important recharging and resetting step. What do you think is the molecule that can recharge our wasted myosin? It's ATP. An ATP molecule, if available, will rebind the myosin molecule and cause a conformational change so the myosin releases the actin. Think of the T in ATP as standing for terminate, as in it terminates the connection between actin and myosin. The energy from the ATP is then used to reset the myosin head into the cocked position, leaving behind an ADP molecule on the myosin head, and the conformational change so it's ready to bind to the actin binding site again. Repetitions of this cycle lead to repeated cross-bridge formation and shortening of the sarcomere. What happens when calcium diffuses away? Troponin C falls back into its initial place and myosin can't bind the actin site anymore. So relaxation. Now, it would be convenient if all muscles contracted the same way, but unfortunately that's just not the case. The muscle contraction that we've discussed so far primarily describes skeletal and cardiac muscle. Smooth muscle, however, contracts using a slightly different mechanism. The precise mechanism for smooth muscle contraction is probably lower yield than the mechanism for striated, but if you have the time, it's worth taking a gander. Focus on the major differences between the two. The primary difference is that smooth muscle does not employ the troponin-tropomyosin system in the same way. That said, the first few steps are the same. Action potentials fire, cause smooth muscle membrane depolarization, and cause opening of voltage-gated calcium channels, which increases calcium levels within the cell. But this is where it changes. Rather than binding to troponin C, the calcium binds to calmodulin. This calmodulin complex then activates myosin light chain kinase, which, as you can likely figure from the name, will directly phosphorylate myosin change. When myosin is phosphorylated, it will form cross bridges and cause contraction of muscle as it moves along the actin molecule. To counter the effect of MLCK is, of course, myosin light chain phosphatase, which dephosphorylates the myosin molecule and causes relaxation. You can think about smooth muscle contraction as a battle between this kinase and phosphatase. In the presence of calcium-activated calmodulin, the kinase efforts win and cause contraction. Interestingly, calcium is the primary signal for muscle contraction, but there are other signals that can cause muscle relaxation. One of the most important ones is nitric oxide, which can be formed endogenously in cells like endothelial cells and can lead to blood vessel smooth muscle relaxation, as in vasodilation. So how does it work? The nitric oxide activates the enzyme guanylate cyclase, which increases levels cGMP. These increased cGMP levels cause inhibition of MLCK. This means that MLCP-mediated relaxation will win out over MLCK-mediated contraction. If you want to review this concept later in greater detail, you should return to the cardiovascular chapter, where we also discuss the use of nitrates in antihypertension drugs. Time for a flash quiz. What type of muscles allow us to sit with good posture? Type 1 slow twitch. Muscle is great and skin is great, but what about bone? Bone is pretty great too. There are two processes of bone formation: endochondral ossification and membranous ossification. What is endochondral ossification? Take a fracture, for example. Cartilage is laid down as a precursor for bone growth, a big fracture callus. You start with a cartilage model, or an outline. Then you convert that into woven bone, or a first draft of the bone. While woven bone is actually bone, its fibers are fairly haphazard and lack strength. However, woven bone is then converted into lamellar bone, or the final draft, which is more structurally and mechanically sound. So what kind of bones form from endochondral ossification? All of the long bones in the body, bones like the femur and the humerus. The other process is membranous ossification. What's missing in this process? The cartilage modeling. Woven bone is formed directly and then later remodeled into lamellar bone. Which bones utilize membranous ossification for formation? Flat bones, like the skull and facial bones. Now that we've talked a little bit about how bones are formed, let's bone up on the different unique cells that they're made of. Osteoblasts are builders. They secrete collagen, the protein matrix around which the mineral portion of the bone mineralizes, and also help catalyze this process. They're derived from mesenchymal stem cells. What can osteoblasts become? They can differentiate further and become osteocytes, which are buried within the bone. Osteocytes make up 95% of bone cells and are the major chemical and mechanical integrators of bone, regulating bone formation, repair, and remodeling, as well as responding to hormonal signals. Osteoclasts have a C, like consume, and secrete HCl and collagenase to break down and resorb bone. They differentiate from circulating monocytes. What happens if osteoclasts are too active? Then too much bone gets resorbed, like in hyperparathyroidism or osteoporosis. What is the classically used marker of the osteoclast on pathology? That would be trap staining. The main thing to keep in mind about PTH is that low intermittent levels lead to buildup of bone. What drug is currently on the market to this effect? That would be teriparatide. Chronically high levels lead to severe bone resorption, as in hyperparathyroidism. Which bone cell do you think PTH revs up? Osteoclasts, causing massive bone resorption and high calcium levels, which you learned in endocrine. Estrogen is also important in bone homeostasis, in that it prevents apoptosis of osteoblasts and osteocytes while promoting apoptosis of osteoclasts. Overall, it promotes bone anabolism. When is estrogen lacking? Either due to menopause or surgery, and osteoporosis can result. One final flash quiz before we move on to pathology. What cells are osteocyte precursors? Osteoblasts.